had a few presentation to go. Anybody an expert? Nick, did you help me with this last time?
Yeah.
I think you did.
Thank you for doing this.
There it is. And we have the full view now, so whatever I did worked. Okay, thanks, Nick. So chapter 19. We're also going to be talking about the structures of chromosomes. We talked a little bit about where chromosomes are located and their contents, but we'll be able to see a couple of pictures of chromosomes that'll help your understanding. We'll talk about mitosis and meiosis. We're going to talk about some different ways that we have genetic diversity and within chromosomes, and why you and your siblings have different genetic makeup, or if you're a twin, why you have a different genetic makeup than your identical or fraternal twin. So just a little bit about chromosomes. Normal humans have 46 chromosomes, so that makes 23 pairs. So when we have 46 chromosomes with 23 pairs, that makes us diploid. It means that we are 2N. 2N. Our sex cells, our sex chromosomes, are 1N. So these are like sperm and egg. We don't want our sperm and egg cells to be 2N, because if 2 plus 2 is 4, we want to end up with a normal human being that has 46 chromosomes. So the difference being that we are 2N, and our sperm and egg cells, they are 1N. Okay? When you have a pair of chromosomes, they're called homologous chromosomes. So there are 23 pairs. So one of those pairs is called a homologous chromosome. The root word meaning homo, meaning same. The autosomes are the 22 pairs of chromosomes that are not your sex chromosomes, so not your X and Y. So 22 out of 23 of those chromosomes are considered autosomes. If you remember the patterns of inheritance, autosomal dominance, autosomal recessive, that's where that word comes from. These are dealing with all of your chromosomes except for your sex chromosomes. The last pair of chromosomes is called the sex chromosomes. These are the XX and the XY. Now there are some genetic variation in diseases here, where maybe there's an extra X chromosome, or there's a lacking a chromosome, so like an XO. And we'll see a couple of those disorders. They're rare, but not impossible, and it happens. And most of the time, people with genetic disorders can live a happy life and maybe have a few complications, but other than that, there are some that are a little bit more detrimental than others, but we'll talk about that in a little bit. A little bit more about chromosomes. So I talked about that haploid versus diploid. Those are those words in red there highlighted for you. Diploid meaning 2N, which means that we have 2 pairs. Okay, haploid meaning that it's 1 pair, or just 1/2 of it, unpaired chromosomes. Those are your sex cells. Your body cells are 46 chromosomes, 23 pairs. So only in the sperm and egg do we see that haploid, and it makes sense. When a sperm and an egg, their goal in life is to the sperm is to fertilize an egg. That's their goal. And if we had 1N plus 1N and they come together, it makes 2N. It makes a fertilized egg with the correct amount of chromosomes that can divide and replicate. So the way we look at chromosomes is a picture called a karyotype. A karyotype is kind of what you see here on the board, and we're able to see the different pairs and their locations. The chromosomes themselves, remember, are tightly wound DNA. So they code for certain things. And so each chromosome pair is a little bit different. As you can see here, each of the pairs look fairly similar, right? They maybe have some variation in their shape or their orientation, but their size is similar. If we had a 1 and a 13 paired up, they would look a little bit different because they're two different locations. And then we have those sex chromosomes, and without looking at the letters, I could tell that this is X and Y because they're two different shapes. If these two were the same shape, or the same size, that would mean to me that they're XX. So this is a male that has the regular amount of chromosomes, 46 chromosomes, and it's a male because it's XY. Females are XX. Okay, so that was a little bit of background on chromosomes. Let's talk about the cell cycle, okay? Everywhere but your nerve and brain cells are regenerating, right? So we started class, maybe what was it, like a month and a half ago. Your skin cells are new skin cells since we first started this class, right? Every 2 weeks, your skin rejuvenates itself. Now, we're not snakes, right? It's not like we shed and it's like, all right, 2 weeks, here we go. It's time to get some new skin. It's constantly happening, right? And they're all at different stages, but the cell cycle is constantly happening, and it's faster than the amount of time that I can explain it, okay? You won't need to memorize, I would say, the cell cycle, but I would understand what each of the stages mean. The next picture will be really helpful in studying for that, okay? So G1 stage, I'm going to kind of flip back and forth between the two. So G1 is kind of that light white yellow color on the left. This is called the growth stage. This is where the cell doubles its organelles and the cell grows in size. So the cell itself, its goal here is to make more cells. And in order to do that, it needs to multiply or grow in size, double the organelles. The organelles are those things on the inside of the cell that are like your mitochondria, your Golgi apparatus, things like that. The S stage, which is the growth and DNA replication, so you're continuing growth and your DNA is replicated. So the exact same DNA that is from the original cell is replicated, because in the new cell, we want some DNA in there, and then eventually that new cell will also go through the cell cycle. The G2 phase, no, this is not Gatorade, it is the growth and final preparation for cell division. Okay, there's proteins in this step that are needed for division, and they are synthesized. Okay, so now we've got like double the DNA, double the organelles. We want to make a new cell from that. So they're going to divide. In order to divide, they have to have some sort of function. And with that function comes proteins. All right, so I think of like protein, we're in the gym, right? They need to be strong enough to pull them apart. Then you have cell division. This is mitosis and cytokinesis. Mitosis is the idea of nuclear division, and this is maybe what you've done in high school before, where like the chromosomes line up in the middle and then they separate and they make daughter cells, and we'll talk about those steps. Cytokinesis is the cytoplasm division, and you can remember that because both of those words start with cytoplasm, cytokinesis. This is the cytoplasmic division. This is everything else besides the nucleus in the cell and how it divides. Yes?
Go back to the slide real quick.
Yes.
How long does that first part take, number one, and all the bullet points under G1, S, and G2?
It depends on the cell. It also depends on the complexity of the cell. So think of like your liver and your kidney. This is going to happen pretty fast. Thinking of your skin cells, takes about 2 weeks. So it just depends on the specialization of that cell. But it is fairly quick. especially because it's an exponential growth. So when one cell duplicates, splits, divides, now you've got 2 cells that can do that. And then now you've got 4 cells that can do that. And now you have 8 cells, and it just is like an exponential growth. So it's fast in a sense that once one cell does it, we have more cells doing it. So there's a couple ways to think about this. There's like you living as your person right now, and then there's like embryo fetus also having to go through these cell cycles to develop normal, body things like hair, fingernails, a heartbeat eventually, right? All those things. Okay. Most cells are in the interphase section, which is those first steps that Jared asked about. Okay, so this is interphase. If you were to look at a cell underneath a microscope, you are most likely like 60% of your cells are going to be in that first stage. Then we go through mitosis. The opposite of mitosis is meiosis. Okay, so we've got mitosis, M-I-T, and meiosis, M-E-I. And we're going to talk about both of those. And the way that I remember the difference between them is because mitosis is my toe, like my toe on my foot. How was my toe made? It was made from mitosis, regular autosomal cells that are dividing and replicating. Meiosis, this is bad grammar, how was me made? How was I made? I was made from a sperm and an egg cell that were fertilized and then cells reproduce. But the division of sex cells is meiosis. And our goal with meiosis is to make haploid cells, right? We want those 1N cells to be able to come together to make a 2N organism. So we have the different stages of mitosis there, and we'll break them down. In order to control the cell cycle, we have certain checkpoints. These checkpoints are that G1, G2, and the M cycle. It's kind of like at the airport when you go through different stages. You're like, okay, first I have to go to the check-in desk and I have to check my bag, and they have to weigh it, and they probably print out your ticket, even though that's like kind of not even a thing anymore because you can do it all on your phone. So you've got a couple of different checkpoints at the airport. Then you go to your security, and then you go to the gate. You don't just walk onto the airplane, right? So these are kind of like different security checkpoints, right? The first one is called G1. And if you pass that one, the cell is committed to divide. Or you could look at this in like steps of a relationship, right? You're dating someone, you're like, man, if we've dated for this long, I'm going to commit to this person, right? We've committed to divide. Then you've got G2 cycle, which pauses to verify the DNA replication. So once we get to G2, we're like, okay, this next checkpoint, we're going to make sure that our DNA was replicated. We want to make sure that this is, we've got enough DNA to make a new cell so that that new cell has the DNA that we need. Then you have the M cycle, which it pauses to verify that the spindle assembly and the chromosome attachment. And we'll see a picture here in a couple of slides. There's also one in your book. When I see the word spindle, I think of spindle fibers because that's what it is. And the spindle fibers kind of look like webbing, not really webbing, but kind of like strings. I think of also like the strings on corn, that little stringy stuff you pull off when you're shucking corn. These spindle fibers allow for the chromosomes to be pulled apart. And we'll be able to see that in a couple of slides here. But these are the different like security checkpoints like at the airport. And once we get through these checkpoints, then we can go into mitosis. So here's that diagram again. Most of the time cells are in that interphase part. Okay, they're hanging out in interphase. Interphase is that purpley, bluish, more dark purple arrow around the outside. That's where most cells are found. Okay, once we hit all the checkpoints, we've gone through G1 and G2. Now we've got M, which is between metaphase and anaphase. And this is a checkpoint to make sure that everything is aligned properly so that we can divide. We got those 3 checkpoints. So we're still talking about mitosis. So what do chromosomes look like while they're in mitosis? What is their structure? Okay, so we've talked about that chromosomes are made up of DNA and protein. The protein is called chromatin. The chromatin is it's essentially just DNA and protein that's mixed, kind of. It's a structural component. The chromosomes that are dividing are made up of two identical parts called sister chromatids, okay? So you kind of, when you look at this next slide here, I'll go back. The sister chromatids are like the X, okay? It's like two parts of the X. You can think of it as like they're sisters, right? The X is sister chromatids. The pair, if we had two X's next to each other, those are homologous. Okay, so maybe I should draw that so you guys can see that too. So the one individual part of the chromosome, okay, kind of looks like that. You have a centromere here in the center. Okay, this is a chromosome. Okay, then if you have both parts of the chromosome, it kind of looks like an X. And each of these are considered sister chromatids. And then if you have two of those, these are called homologous chromosomes. Okay, let me catch the Zoom group up to speed on that. So I drew this picture essentially on the board, and then what I added was when we have two chromosomes, two sister chromatids, two pairs of sister chromatids, these are considered homologous. And I'm just going to write homo L so I don't have to write the whole thing. That's called a homologous chromosome. Okay, so that last one that we didn't get to on the previous slide is called a centromere, and that's that kind of circle that I drew on that first one that holds the chromosome together. Okay, I kind of think of it like a push pin, you know, when you hang a picture up on a corkboard, I think of that as what a centromere is. And again, on that last side, we have a chromosome. When a chromosome is replicated, it becomes sister chromatids. When we divide those chromosomes, it becomes just a single chromosome. The spindle in mitosis, so the centromere, the microtubule organizing center of the cell, so that's that little dot, that's the push pin. Then you have the aster, which is an array of microtubules at one of the poles. So essentially there's like some strings coming off of either side of the cell, and I'll show you here in that next slide. Those are those strings. So you see the chromosomes down the middle, those strings that I keep drawing with my hands, those are those spindle fibers in mitosis. And then you have centrioles. These are short cylinders of microtubules that assist in the formation of the spindle fibers. Okay, so the goal of the spindle fibers in mitosis is to pull it apart and divide. You can think of it as like webbing for spiders or something, pulling it apart. Because our goal here is to replicate DNA, split that, so we have two cells, right? So here we have those centrioles in the centrosome. You have your chromosomes that line up in the middle. Those spindle fibers are those string-like looking things that are attached to the centrosomes. And then you have the aster, which are the little cylinder type things around the outside. So those are the spindles of mitosis. All right, there are 4 phases to mitosis. Remember, mito, dividing autosomal cells. This happens from an embryo to a fetus and forward. This also happens pretty much daily in your own bodies, okay? And like it says, it occurs for body cells, skin cells, liver cells, kidney cells, hair cells, things like that, all of your body. This is not your sperm and egg cells. It is used for cell growth and repair. So it's the same thing. You get a cut on your hand, you're going to eventually have a scar, maybe. Hopefully you do or do not, whatever that is to you. But the way your skin rejuvenates and heals from that is by the cell division and replication. A diploid cell, so the cells in our body, we make and divide an exact copy of the nucleus. Okay? So this is kind of like you're taking those skin cells to the copy machine and you just hit copy. And it's like shooting out copies, identical copies, okay? Meiosis is where we start to get a little bit of that genetic diversity. So if you were asked the question, compare mitosis to meiosis, mitosis makes identical copies, right, the copy machine. Meiosis is where we have some genetic diversity, which is why if you have two biological male and female humans that reproduce and make offspring, why if they have another offspring, the same two people, that offspring is different than the first offspring. It's because meiosis is the reason we have genetic diversity. We'll talk about that. So here's an overview of mitosis, okay? That top picture is just showing you where those chromosomes are located. They're located in the nucleus. The nucleus is that purpley kind of thing at the top. Okay, we have a centrosome and a centriole. It's showing that at the top, these are 2N. So that means that we have 4 chromosomes here, or 2 pairs. All right, you can see that there's a blue and a red, and there's small and big. All right, there you guys see that? There's 2 red ones, 2 blue ones. One is small, one is big. Now your real chromosomes are not blue and red, but this is just going to help you show you how they pair up. So the first step is DNA replication during interphase. Okay, we want to make an exact copy of DNA, so this new cell that we're making is exactly the same. It is duplicated chromosome consisting of 2 sister chromatids. Right? Remember what a sister chromatid is? It's the X, it's each part of that X. Then you have division. So you made the exact copy, and then we divide, and now each of the daughter cells, these are considered daughter cells here at the bottom, look exactly the same way when we started. They have 2 red, 2 blue, 1 big, 1 small. The first step of mitosis is prophase. This is where chromosomes condense and become visible. The reason they become visible is because they are condensing, okay? Visible just meaning to, like if we were to look at them through a microscope, okay? When you look at interphase, where the cell normally is, if we were to look under a microscope and see a cell that's in interphase, we couldn't see the chromosomes when it's just hanging out. When we start mitosis with prophase, we can start to see the condensed and visible chromosomes. A way to remember this stage is like if you're a pro at something, like you're a pro soccer player, you're a pro violinist, you're a pro artist, I think you can become more visible, right? Think of all the professional people out there, doctors, things like that. They're more visible to the public eye. That's the way I remember this section of mitosis. Prophase, the first phase, those chromosomes are condensing and becoming more visible. The nuclear envelope fragments. So when you have a fragment of something, it just means you have like a part of it, or there's it just kind of comes apart a little bit. So that nuclear envelope essentially is the envelope that's around the nucleus. It fragments. The nucleolus disappears, and the centrosomes move to opposite poles of the cell. I'm going to show you in the next picture what that looks like. Those centrosomes, which are going to aid in those spindle fibers that are going to pull that cell apart, they go to opposite poles of the cell. Then we can start to see the spindle fibers appear, and they attach to the centromeres. I think it's a lot easier if we look at this. This is a real cell up here at the top. You can see the aster, you can see the chromosomes a little bit. They're starting to become more visible. That's what that middle part is. I'll show you guys here. Those are those chromosomes. That's the aster, so we start to see those spindle fibers. And here we start to see the duplicated chromosomes for prophase. There's two steps to this, early prophase and actual prophase, but just worry about prophase in general. Next, we have metaphase, and this is when the chromosomes line up at the middle of the cell, the equator. Spindle fibers become fully formed. All right, this is another picture here. You can tell that this is metaphase because they start to line up in the middle. And I can kind of draw that picture here. This is where the chromosomes line up in the middle. I do not know if this is a good example, but do you guys know who Meta World Peace is? Meta World Peace, have you ever heard of that person before? I am not an expert on that person, but when I think of Meta, I think of Meta World Peace. And world peace would be if we all could just line up in the middle, right? We could figure out a way to just come together, right? So that's kind of what I think of Meta, is like Meta World Peace. We're coming together in the middle for those chromosomes. All right, then we have anaphase. These are where the sister chromatids separate at the centromeres and move towards the poles. They are not separated yet, but we start to have a little bit of some division here. They're not divided, they're in the middle of division. So the chromosomes you can start to see separate here, but they are not separated, right? So you can see where they're being pulled apart. The sister chromatids are being pulled apart, but they're not actually separated yet. I don't really have a great way to remember that one, so if somebody else can come up with something, that would be great. Mitosis, telophase, and cytokinesis. So this is where the chromosomes arrive at the separate poles. We have something called the cleavage furrow, which is essentially just kind of like that space in between, that they're not separated. Like I said, they're not entirely separated yet, but they are having some sort of like shape change here. The nucleoli reappear, the spindles start to disappear. So those things that helped us pull, those strings that helped us pull them apart, they're disappearing now. The two daughter cells are formed by a ring of actin filaments in that cleavage furrow. So the way you can tell of telophase is they start to divide. You can think of this like a telephone. You know, we have those in our pockets, right? Telephone, you can like call somebody who's on the other side. That's kind of how I think of the two are at different poles, two telephone poles. All right, here's just that overview. Oh, sorry, I thought we were still talking about mitosis. Does anybody have any questions about mitosis? I usually get the question, so what do I need to know here, right? This is a lot of information. What I would say is you should know the order, right? You should be able to say prophase, metaphase, anaphase, telophase in order, right? And that's also in the PowerPoint. And then just know what each stage does. Okay, I probably won't show you a picture like this and you'd have to identify something in the picture. So just be able to identify what those, or more essentially define what each stage does and maybe in the correct order. So now that we talked about mitosis, let's talk about meiosis. So meiosis is the nuclear division that occurs to make 4 haploid cells, okay? In mitosis, we make 2 identical daughter cells. They're identical, they're the exact same, and we end up with 2 of them. With meiosis, we end up with 4 haploid cells, and there may be some genetic diversity here, so they may not be identical, okay? It is used to make gametes, which are also another word for sperm and egg cells. Like it says, it occurs in the sex cells, and it has 8 phases. Holy cow. So what we end up doing here is the first half of meiosis looks like mitosis. It's pretty much identical. The difference between meiosis and mitosis is meiosis has 2 divisions. All right, you can see the first division and it looks very similar to mitosis. Because it has a second division, it essentially divides it again and makes them haploid to make the sperm and egg cells. So these are your sperm and egg cells at the bottom here. And there are two divisions. Your first division is here. Your second division is here. And I will show that here on Zoom. You guys have a first division here and a second division here. So there's two steps to meiosis. We've got meiosis 1 and meiosis 2. The first meiosis is similar to mitosis. We have prophase, metaphase, anaphase, telophase. They are indicated by 1 or 2 because there are some prophase, metaphase, anaphase in meiosis 2. So prophase 1 does exactly what you think it does. Those homologous pairs, crossing over occurs here in which there's an exchange of genetic information. We'll talk about what crossing over is. There's kind of some like mixture of genetic information so that we can have some genetically different daughter cells per se. We have metaphase, which is where they line up at the equator. They line up in the middle, those chromosomes. Then you have them where they start to separate and move towards the opposite poles. Then we have two daughter cells that result in 23 duplicated chromosomes. So pretty much that's the same as mitosis. All right, but mitosis is not the goal here. Here's that prophase 1. It's where those chromosomes condense, become visible. Crossing over occurs. We'll talk about what crossing over means. Our goal here is then that we make a diploid cell. So from prophase 1, we go to metaphase 1. This is where they line up in the middle, meta world peace. Then we've got prophase 1, metaphase 1, anaphase 1. Anaphase is where they start to separate, but they are not separated. They move towards those poles. Then we have telophase 1, which is at the bottom, and we made two daughter cells. The goal for meiosis is 2 divisions and 4 daughter cells. Mitosis is 1 division and 2 identical daughter cells. So meiosis 1, then after telophase 1 here, so this first part here, very similar to mitosis. Now we have interkinesis, which is where the chromosomes still consist of 2 chromatids. So now we're going to get ready for that second division. We want to make another separation so that we have 4 haploid cells. Okay, but before we move any further, it's important that I talk about what crossing over is. Okay, that's happening during that prophase. It's where there's an exchange of genetic information between non-homologous sister chromatids during synapsis. Okay, so what do those words mean? Genetic information, meaning maybe a few nucleotides are kind of exchanged and they're kind of going back and forth. These are happening between non-homologous sister chromatids. So you've got an X over here and an X over here that are 2 different, and they're crossing over to each other. They're kind of showing that this is a way that there's we can share some genetic information. Crossing over is the reason we have genetic diversity. So this is why it says that the increases the genetic variation or the variety of the combination of genes in an order there. Here's a picture that'll best explain it. I think of these kind of like pipe cleaners. You guys remember pipe cleaners from like crafting? If you were to like snip a blue pipe cleaner and glue it to a red pipe cleaner, that's kind of what you have here. So if we have a chromosome that has A, B, C, D genes, and then you have another chromosome that has lowercase a, B, C, D, those are the sister chromatids. That's the X. And then there's another one over here. We're crossing over between the chromatids of 1 and 3 allow for the result. The result is on the far right here. And now you have some pipe cleaners that are like all red, all blue, a little red and some blue, and a little blue and some red. Okay, so that's how we get genetic diversity, is through crossing over. If the question asks, why do you have different genetic makeup than your siblings or another person in your class? One of them could be, the answer would be crossing over. The other could be independent alignment. Independent alignment is where it's during metaphase 1. It's where homologous pairs align independently at the equator. Okay, it gives rise to various possible combinations of maternal and paternal chromosomes and gametes that descend from this cell. It's how they're oriented before they split. Okay, so the word kind of gives it away. Independent. When somebody is independent, they kind of do their own thing, right? Miss Independent. That's why I love her, right? No. So we have you line up in the middle, and if they're kind of aligning how they want to, instead of exactly the same like they are in mitosis, we're going to have some different alignments, and when they get separated, we're going to have different results, okay? So here are some just different ways that these exact same chromosomes could line up differently and have different results. All right, so the first one we've got big blue, little blue, or like medium, small, big blue, medium, small, big red. If you look at the next one, it's a little bit different. It's medium blue, small blue, big red, medium red, small red, big blue. You see how it changes here? And the next one's another option, and then another option. So it's saying that the way they line up at the equator in the middle can be a little bit different, right? You can also think about this as politics, right? We've got some red, some blue. Yeah? All right. Meiosis 2 is the second part of meiosis. So we already did the first part of meiosis, and we have a division. We've got 2 daughter cells. Now we'd like to divide again. We want to make haploid cells. We want to make the sperm and egg cells that have half the number of chromosomes, so that when they go and get fertilized together, we're making a normal human that has 23 pairs, 46 chromosomes. So this phase, it also helps that they are listed with the number 2 next to it. And the phases seem to have some common themes here. So prophase, remember, they condense, they become more visible. That happens again. Metaphase, the chromosomes align at the equator. Anaphase, sister chromatids separate to the opposite poles. Telophase, 4 daughter cells result. So we now split. Remember, this is happening at 2 cells, because the first phase of meiosis, we made 2 daughter cells. Now with these 2 daughter cells, they are dividing again. So now we have 4 daughter cells that each have 23 unduplicated chromosomes. So they're just 23, 23, 23, 23. And when they come together with the opposite, either a sperm or an egg, they make 46 together. So here's a little image about prophase 2. Those chromosomes of those 2 daughter cells condense and become more visible. Then you move on to metaphase 2, which comes after prophase 2. This is where they line up in the center. Do you see now in this picture, there was some crossing over. Now we did start with a small blue and a big red with the one on the left here. And now if you look real close, I'll try and zoom in for the people in class here. Oh, wow. I'm just getting ahead of myself. So much to learn. Okay. Here we go. You guys see how the blue and the red kind of mixed a little bit? Kind of like they dyed the tips of their hair. Just a little bit different. That's where that crossing over happens. Crossing over is not a bad thing, right? It just happens. It's how we get kind of a mixture of the same genes. It's also why, while you and your sister or you and your brother, you don't look the same, but you're like kind of the same. You've got some features. It's the same thing here. You maybe have some features that are very similar, but you're not the same. Okay, that's metaphase 2. They line up in the center. Then we have anaphase, where they start to separate. Remember, this is happening with 2 daughter cells at the same time, because we already split once. Now we're splitting again. Our goal here is to make 4 genetically diverse daughter cells that have half the number of chromosomes. Telophase, you guessed it, that those spindle fibers disappear. And we start to have that separation there. We have that cleavage furrow down the middle. And our result is 4 haploid daughter cells. Let me say that one more time. The result is 4 haploid daughter cells. Meiosis, similar to mitosis in a sense that we you know, we can see the chromosomes, they're condensed and visible, they line up, they separate, and we've got some division, okay? But in meiosis, we do that twice, and we end up with haploid cells. All right, when we produce sperm and egg cells, the process of making a sperm cell is called spermatogenesis. To generate, essentially, is what the second half of that word means. For males, this is a continual process after puberty. It's also the opposite with females because there comes a certain time in a female's life where they're unable to produce eggs or have a reproductive cycle. Where males, this process of making sperm happens throughout their life after puberty. About 400 million sperm are produced per day. Okay. If you think of the opposite of that, now the oogenesis, kind of the way to say that, is the process of making eggs in females. During meiosis, one egg and 3 polar bodies are formed. Polar bodies act to hold discarded chromosomes and thus disintegrate. Normally 1 egg per month is produced, which makes sense, right? And about 500 are produced in the entire life, right? So we've ever heard somebody say like, oh, time's a ticking, right? We gotta go have a baby or something. Females only have a certain amount of eggs in their body, and they are born with those eggs. That's just how many they have and the possibility. All right, so here's kind of a diagram of that, the production of sperm and the production of eggs and how they are compared here. You can also see in the bottom picture there the egg and the sperm together, fertilization. And then the fusion of the sperm and the nucleus with the egg nucleus. That fusion that happens between the nucleus of the sperm and the egg, that is called fertilization. All right, so let's compare, right? This would be a pretty good test quiz lecture assignment question, right? Tell me the difference between mitosis and meiosis, right? What are the differences? First, what's their function? Mitosis is for growth and repair. That's how you say that together. It's Greer. Growth and repair of cells. So this is your body cells. This is cell division from embryo to fetus when the sperm and egg cell are fertilized and they start cellular division and replication. And then you have meiosis, which is the formation of gametes. This only happens in sex cells, which is the next bullet point. Mitosis happens in all of your body's cells except for your sex cells, and meiosis is just in your sex cells. In mitosis, we have 1 division, where in meiosis, you have 2 divisions, which makes sense because we want to make haploid cells with meiosis. We want to make diploid cells with mitosis. We make genetically identical diploid daughter cells in mitosis. I'm going to say that one more time. Genetic identical diploid cells in mitosis, and genetically different haploid cells in meiosis. This is a pretty good slide here. Another way, if you're a more visual person as opposed to bullet points, is these pictures here. So mitosis only has 1 kind of flow, right? You go prophase, metaphase, anaphase, telophase, 2 identical daughter cells. Meiosis kind of has 2 stages. You have 2 divisions. You do go prophase 1, metaphase 1, anaphase 1, telophase 1, 2 daughter cells. Then you do it again. Prophase 2, metaphase 2, anaphase 2, telophase 2. 4 haploid, genetically diverse daughter cells. Here's the second half of that. So this is what the result of those last points were here. Mitosis, we've got those 2 identical daughter cells. Meiosis, you have those 4 genetically diverse haploid cells. Okay. Now, this happens a majority of the time, but there's also some time when there's maybe a mix-up, a mistake, and there's nothing that anybody can do to prevent it. It just is a fluke thing. And these are when there's some changes in the chromosome number. And maybe the most common is Down syndrome, right? When somebody has an extra chromosome. There's some other diseases where you're like missing a chromosome, and that would be where there's only one copy, like Turner syndrome. And like I said, it doesn't mean that the person is incapable of having a life. So it just means that there may be some developmental differences. And some genetic diseases are different than others based on their location and how severe it is. All right? So non-disjunction. When I look at the word non-disjunction, if I break that up, non-dis, both of those words to me mean something's wrong, right? Something's off here. That's the way I remember this. So this occurs when both members of the homologous pairs go to the same daughter cell during meiosis. So normally they would divide, and there'd be two diploid cells with some genetic diversity. Well, with non-disjunction, they divided, but they went to the same cell. And I'll show you guys in a picture here in just a second. So when that happens, our chromosomes are going to divide differently, and one of the cells is going to be extra, and one of the cells isn't going to have one. Okay? So the two results that are possible from non-disjunction is monosomy and trisomy. The root words meaning mono and tris. Mono meaning one, tri meaning three, which is the way you can tell these apart. So from non-disjunction, when they didn't split properly, it's possible that your cell has an extra chromosome, which would mean that there's 3 copies of that chromosome, which is the example of Down syndrome, where there's 3 chromosome copies at the space of 21 on your 21st chromosome. Then you have monosomy, which is 1, which means that because they split differently, one cell got kind of left out there, which means that you're missing a chromosome in that space. This is called Turner syndrome, where there's only 1 chromosome.
And can that chromosome be any of the 22?
Yes, it can be. There are some that are more common than others that have where non-disjunction happens more common. That's why like Down syndrome is a 21st chromosome, and it happens a lot, not all the time, but a lot, and that's why we know a lot about it. Turner syndrome, it's out there, but you probably haven't heard much about Turner syndrome. So it's the same thing. So it can happen on any of the 23 chromosomes, but there's some that are more common than others. It has to do with the size. It has to do with the functions of what those chromosomes do. It also has to do with evolution. So that's an interesting one right there. Okay, so non-disjunction. Remember, non-dis. Okay, something's going wrong here, right? We've got 2 negative prefixes. There we go. So non-disjunction, it can happen at either of the divisions in meiosis. So it can happen in that first division, right? Because we have 2 different divisions. You have a division here from 1 to 2, and you have a division here from 2 to 4. Okay, so non-disjunction can happen at either of those divisions. And when that happens, as you can see on the left-hand side, you have ones that end up with 3 chromosomes, and you have a couple that end up with 1. And then on the other one here, you have some that end up with 2, some that end up with 3, and some that end up with 1. It just depends on when it divides and when the non-disjunction occurs. So normally, females have 2 X's and males have an X and a Y, okay? Males do have 1 copy of an X, right? They are XY, so they do have an X. In female embryos, 1 X chromosome can become inactivated, and it's considered a bar body in each cell. The other way to look at this is that there can be a change in that chromosome number. This is just showing the example of Down syndrome here, which we talked about briefly. Okay, there's an extra chromosome, and you can tell that because of that karyotype in the middle there. There's 3 chromosomes at that location. Okay. It means that they have extra copies of whatever that gene is that is in that location. Turner syndrome is located at the sex chromosome. This is when there's no second chromosome, whether that be a Y or an X. And when you have Turner syndrome, some symptoms or effects of that, you have a short stature, you are broad-shouldered, there may be folds in your skin and your neck, and you have underdeveloped sex organs and breasts, which like kind of makes sense if you think about it. If you're lacking one of your sex chromosomes, the underdeveloped sex organ seems like a fairly, it seems like explanatory with that if you're lacking a chromosome in that sex cell location. The opposite of that is when we have an extra X chromosome in that sex cell location. So you have an XXY. This is the Klinefelter syndrome. This is where you also have underdeveloped sex organs and breast development. You may have large hands and long arms and legs. So this is where in that chromosome location, you have an extra chromosome. Poly-X females, the name kind of gives it away. So poly meaning many. So if there are more than 2 X's in that location, they're considered poly-X females. They tend to be tall, thin, and do not usually have, I don't like that word, mental challenges. And then XXXX have severe mental challenges. So when you have a little bit more, that is uncommon, but you will have some mental challenges there. Jacob's syndrome, probably named after a guy named Jacob who had this, is where you have an extra Y chromosome in that location. So these are some of those different genetic diseases that happen with the variation of the chromosome number due to that non-disjunction, some variation in the way that they were split. If somebody has Jacob's syndrome, they may be tall, they may have persistent acne, they have speech, and maybe some reading process issues. Okay. There may be some changes in the chromosome structure. Okay. If there are changes in the chromosome structure, there are some ways in which that would happen. All right. You could have deletion, which is essentially like when you type something wrong and you're like, oh, delete, delete, delete. That's the loss of a piece of a chromosome. If you have a chromosome that is like this, you would have Williams syndrome. You have translocation, which is the movement of a chromosome from one segment to another. If you think of the word translocate, maybe somebody might get translocated for their job, right? They may have to move to a certain different location for their job. That's the same thing here. You have a part of a chromosome that moves to the other. And I'll show a picture on the next slide if you're more of a visual learner. Duplication is essentially what you think it is. If you duplicate something on your computer, right, or you make another copy of, this is the presence of a chromosome segment more than once in the same chromosome. So it's like copy, paste, 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 right? That's duplication. Inversion is a segment of chromosome which is inverted 180 degrees. So while it looks like this, it was inverted 180 degrees and now it looks like this, if I can bend in that way. So here's some pictures of that to kind of give you a visualization. Deletion, duplication, inversion, translocation. This is if there's a change in the actual chromosome itself that is indicating a genetic disease or disorder. So we can have non-disjunction be a reason there's some genetic diversity and disorder because they split in the wrong way, or a genetic disease or disorder can happen from the actual structure of chromosome itself. Maybe there was a deletion of a part, or a duplication, too many copies, or an inversion where something was flipped, or a translocation, something was moved to a location that it wasn't supposed to be. The next slide just shows again what that chromosomal inversion means. So you have a region where they do crossing over, and remember, crossing over is a good thing, right? We want genetic diversity. Can you imagine if we were all the same? That would be really boring life, in my opinion. But if we were all the same, we don't want that. We want genetic diversity. So crossing over is good, but inversion is not, right? We don't want it to be backwards. There could be some defects for that. And then on the last one, it says duplication and deletion were happening there in both. You can kind of look at when you go back to study this, kind of look at where you start and the order of the letters, and then look where you end, right? So we went A, B, C, D, E, F, G, uppercase, and then A, B, C, D, E, F, G, lowercase. And then on the right-hand side, it says A, B, C, D, E, B, A. And then some of them are capital and lowercase. And then you have G, F, C, D, E, F, G. Some of them are capital and lowercase. So you're kind of seeing how, because of that inversion and the duplication and the deletion, we've got 2 different chromosomes there. And that means that they're going to code for different functions. Remember, that's what DNA is for, is to code to make proteins. Proteins help with functions of our body. And that's where we have some developmental issues or physical changes. Lastly, here we got changes in chromosome structure. Same thing, it's just showing that like your H can be lost there, and that would be considered a deletion. So on that far left, you have A, B, C, D, E, F, G, H. And then on the next one, you have A, B, C, D, E, F, G, and the H is lost. Translocation is where that Y and Z and the G and H kind of flip. They're translocated. They went to a different spot. All right. 54 slides in an hour. That's how it's done. Does anybody have any questions over anything that I went over? There was a question about outfly, and I'm just going to answer it for everybody, just to be clear. I'll make sure to email you to communicate about that. So if outfly were to happen on a day that we have normal class, that would get adjusted. It pretty much would mean whatever we were supposed to have that day would happen the next day. So if it happens on a Wednesday, like 2 days from now, they're like, it's outfly. I think we meet on Thursday and we do lab then on whatever. It's like essentially that day just doesn't exist, right? And we just go on to the next one. But I'll make sure to email you guys and communicate it very clearly so that we're good on that.
Well, actually, if we have outline on Wednesday, our Wednesday class gets moved to Friday.
That's what it is.
Yeah, so
you're right.
we wouldn't use per se if it goes on Wednesday, you could have really severe Tuesday, Thursday class. Tuesday, nothing on Wednesday because outline, and then Thursday again would be Tuesday, Thursday class.
Yep. And but if it happens on a Thursday, we would not have class on Friday because the things that were on your Thursday,
The Thursday class is moved.
yeah, you guys got it.
Would we do lab on Friday if class on Wednesday is outline?
Yes.
So we just plan to move.
Whatever we were supposed to do, just like push it to the next day. Whatever happens. Also on Zoom, I'm going to pick on Sam. Sam, do you guys have something going on tomorrow that you wanted to share?
Yeah, I do. We have something going on. Yeah, the soccer team is hosting a Unity Day. It's like a Unity demonstration. It's 11:30 a.m. at outside Old Main. Obviously, wear a mask. It's socially distanced. But the soccer team thought it was important to show the unity in the campus as well as the Waverly community. So if you guys have time from 11:30 a.m., it's only going to be like 15 minutes. It's going to be a couple speakers and a couple other groups out there talking about next steps and stuff. And I'm one of the speakers, so I'd love to see you guys out there.
All right. Is that something that interests you? 11:30 tomorrow at Old Main. Other than that, if you're in person, please wipe your table down. Thanks, Sam. Zoom, you are free to go. And I will see you guys when I see you guys.
You have this. Docs.